सो पार्ट वन में मैंने कवर किया था बेसिक कंसेप्ट्स। इस वीडियो में मैं फेट मैप की बात पार्ट टू स्टार्ट कर रहा हूँ। हियर यू कैन सी, ये डायग्राम से आप देख सकते हो द फेट मैप। So this one is of zebrafish, this is a frog. So the anterior portion, this is the posterior. So this is the epidermal, this is the neural ectoderm, this is mesoderm, and this is notochord, and this is endoderm. And the yolk is present here, you can see. This is the fate map of early gastrula stage. But here you can see the mouse or chick fate map. So this whole green thing is epidermal ectoderm. This is neural ectoderm. The notochord at the center, mesoderm after that, then endoderm, then extra embryonic mesoderm. So there are many various techniques which can be used to generate the fate map. So the first one is direct observation of the living embryo. So here some embryos we can see and can relatively quantify them. And in some embryonic cells, the naturally cytoplasm has different colored pigments. Here you can see the yellow colored pigment is present in the cytosol. Then it leads to the muscle formation. Then second one is the dye marking. You can use the dye, like apply vital light to the region of the interest. So you can use different types of dyes for different layer, like for ectoderm green, mesoderm blue. And they are also fluorescent dyes. So when you see them in light, then they can fluorize, that they can glow. They are examples of this type of thing, like fluorescein conjugated dextran. And for genetic labeling is permanent way of labeling. So here see chick quail chimera. So the grafting of the quail cells in chick embryo region of interest. So here you can see the quail cells are grafted in the chick embryo. When then at the last, upon development, the full grown chick embryo. You can see the coil cells. The coil nucleus has a heterochromatin DNA concentrate around the nucleoli, and cells specific antigen expression is there. Coil cells that expresses coil specific antigen on the surface. Chick cells express chick specific antigen. And this one describes the frog development. You can see. Then the commitment, ectoderm or mesoderm, which layer? So cell lineage fix or cell fate fix. Cell is determined for particular fate. So A cell, B cell, they can have ability to form any type of cell. And this is undifferentiated cell that is not specified, that it can form into any type of cell. So our lineage ectoderm can form from here, and the B lineage, the mesoderm, form from here. So the before commitment, if we change the cell position, cell's fate can change. After commitment, if we change cell position, cell fate cannot be changed. You can remember this. Then commitment involve two stages: specification, that is early stage, reversible; then determination, final stage, it is non-reversible. Specification is early stage of commitment and it is reversible or labile phase. If we change the cell position, cell fate can be changed. The specified stage cells is capable of differentiating autonomously in the neutral medium. Here you can see the cell A and cell B. Here it remains undifferentiated cell and it forms the ectoderm. So it is not specified and it is specified. Determination is the final stage of commitment and it is irreversible, and we change cell after determination, then its fate cannot be changed. Then specification is of three types: the autonomous specification seen in invertebrates, annelids, tunicates, mollusks; then conditional specification, vertebrates, amphibia, reptile, birds, mammals; and syncytial specification in insects. Then see another one, the autonomous specification, if we can talk. So the signal is present within the cell cytosol, and here you can see inside zygote there are cytosols, the mRNA-derived proteins, and the blastomere inherits it is sets of the transcription factor, and it acts as a transcription factor. So early stage of embryo, then up to 8 cell stage, and little or no gap between specification and determination. Then potency of the cell. If we change cells position, cells fate cannot be changed. And here you can see the developmental pattern: zygote to 2 cell stage to 4 cell stage, then 8 cell stage. Mosaic development pattern: each and every cell required for complete embryo formation. For mosaic development. And if we remove some cells from embryo, the adult lack this structure. The mosaic pattern of development, you can see ectoderm, notochord. Then the macho gene regulates the muscle development in the tunicate. The macho gene product is muscle differentiation in tunicates. So inside egg is the macho transcript, which upon its cell stage formation also it is present as a macho transcript. And you can see the process here. And this is the tissue transplantation experiments. Many questions came from this. If the donor cell is ectoderm and host cell is endoderm, then what is the cell fate? It become ectoderm. If the donor cell is endoderm, host region is ectoderm, then it form the endoderm. Like this, you can see in early gastrula and late gastrula, this is different. Then conditional specification. Example in vertebrates, reptile, birds. Signal comes from outside, that is from neighboring cells, and cells interacting with the neighbor cells. Then there are paracrine interaction and juxtacrine interaction. This is all the stages from zygote to late gastrula. Cell potency, developmental pattern, then drift demonstration of conditional specification. It is very important. So the observation of the driss is that complete small size pluteform and generalization of all type of cells of the pluteus lava by single cell and non-identical pluri developed. Conclusion: conditional specification present, prospective potency is greater than prospective fate, loss compensation is present in sea urchin harmoniously equipotent system. So that's all. Thank you everyone.